Hello and welcome to the Dog Histology Laboratory. Now, as you probably know, histology is the study of tissues, and tissues are groups of cells that are joined together to perform specific functions. Now, most of you that are enrolled in biology or anatomy classes have probably spent hours and hours looking through the microscope trying to identify the different types of tissues that are on those microscope slides. Well, today we're going to kick that up a notch. We're going to try to identify the different types of tissues that are in tubular meat products, otherwise known as hot dogs. Now, if you ask somebody what is in a hot dog, they'll probably say something like lips and assholes. But in truth, there's other things besides just lips and assholes, and even these things have different types of tissues in them. So let's talk about the candidate tissues that we might expect to see within our hot dogs. Okay, our first candidate tissue here is skeletal muscle fibers. Now, skeletal muscle is what we think of when we think of meat. When you order a steak, you're ordering a big slab of skeletal muscle. And that skeletal muscle is striated and it's voluntary, and we would hope that it would be the primary ingredient in each of our hot dog products. Now, after that, we might expect to see some adipose tissue. Adipose tissue is otherwise known as fat tissue. It's found in the hypodermis of the skin. It helps to store energy and insulate the organism. And when we mix it in with skeletal muscle fibers, we get a really, really tasty hot dog. So we'll expect to see plenty of adipose tissue as well. And finally, our third tissue type here is fibrous connective tissue. Now, fibrous connective tissues are found scattered throughout the dermis and hypodermis, and because skin ends up being in hot dogs and stuff like that, we'd expect to see plenty of fibrous connective tissue in there as well. Okay, now let's meet the contestants. The contestants are, of course, hot dogs. The first of these hot dogs is Nathan's bun-length hot dog. The next contestant is the Ballpark all-beef Angus hot dog. And the third contestant here is the Safeway Organics Angus hot dogs, which are uncured. So all of these hot dogs are pretty high-end, and we'd expect them to have a high proportion of muscle tissue that is skeletal muscle. Now, as a ringer, I'm going to throw in an outside one, and these are Vienna sausages. Now, Vienna sausages, if you haven't had them, are probably the most processed meat product on the planet. If you look at a Vienna sausage, it's not really recognizable muscle fibers or anything like that. And so we're expecting them to have fewer recognizable muscle fibers and maybe a higher proportion of adipose tissue. Okay, so our research question today is which of our 4 hot dog brands will have the highest proportion of skeletal muscle fibers, or meat, and which will have the highest proportion of adipose tissue, or fat? So that is the research question. I'll leave it to you to determine your hypothesis. For example, if you think the Vienna sausages are going to be higher in fat and lower in protein, you would say so. But you might have different opinions based on your experience with each of these tubular meat products. Now, for each of these hot dogs, we're going to pull one hot dog out of the pack, and then we're going to make 3 transverse sections about, half a centimeter in diameter, or in thickness, just like that. So we've got our 3 cross sections here, and we can throw these other parts of the hot dog away. And now for each of these 3 cross sections, we want to take a small little core. What we have here is basically a cork borer. We're going to punch it in there. Okay. And then you can see we have a little bit of that tissue in the end, and we're just going to extrude that into our dish in these pre-labeled wells. So this is for Nathan's hot dog. Okay, finally we're going to do our Vienna sausages. So we pull those out, open that. Look at that in there. Doesn't that just look, ugh, okay. So we'll pull out one of our Vienna sausages. And again, just do 3 little transverse sections. So that cuts quite nicely. And then we're just going to poke those 1, 2, 3. And then we should be able to extrude those. Okay, so we've got our nice little sausage chunks there. 1, 2, 3. So we have 12 different wells. 3 wells are 3 different subsamples of the sausage. We have Nathan's right here. We have the Ballpark on this side. And then we have our Safeway Organics here. And at the very end, we have our Vienna sausages. Okay, now the last thing we have to do is get out some formaldehyde. And we want to be very careful with this because formaldehyde is toxic. And then we're going to take a little bit of formaldehyde and put it in each well because the tissues need to be fixed before we can process them histologically. Okay, so after preserving our specimens, we then put them through a graded alcohol series and eventually a resin series until we dehydrated them, we had them in resin, we eventually embedded them in a resin block that looks something like this. So that contains our hot dog tissue in there, and we're going to section it on something called a microtome. Again, a microtome is just an instrument that we use to cut tissues very, very thinly so we can look at them under the microscope. Okay, once we get a good representative section, we're just going to lift that off. And then we're going to place that on a microscope slide with some water on it and float it on there. And we're just going to make about another 8 to 10 sections to try to get the optimal section of that hot dog tissue. Now, once we have enough sections on our microscope slide, we're then going to take that slide and we're going to place it onto a hot plate so we can dry out those sections. Okay, now that our slides are all dried off, we put them in a rack, and next we're going to put them in a series of stains so we'll be able to see the different tissues underneath the microscope. Okay, now that we've finished sectioning and staining our hot dog tissues, we have photographed them underneath the microscope, and we've posted those images here so you can download them and use them with your class. So if for some reason this link doesn't work, you can just email me at langston@hawaii.edu, and I'd be happy to share the images with you. Okay, so there are 3 different images for each of the hot dog samples. So there's a total of 12 images that have to be analyzed in order to determine which one of these hot dogs has the most fat and which one has the most muscle tissue and other tissues as well. Now, before we do that, I want to talk about the other resources that are available at that link that I just gave you on the previous slide. Now, in addition to the 12 histological sections from our hot dogs, that resource folder also contains some other valuable files. In there, you'll find a scorecard for your hot dog. And the scorecard contains basically our 4 different categories in there, our 4 different hot dogs, and it also has the 5 or 6 most common tissues you're likely to see on those hot dogs. We also have a histology cheat sheet, and this cheat sheet is very important because a lot of the tissues that you're going to see in these hot dogs don't look like the tissues you see under the microscope. These hot dogs have, after all, they've been cooked, they've been chopped up and macerated, and so the tissues are going to look somewhat different. Next up, we have nutrition information for each of the hot dog brands, and this is important because maybe you want to do a comparison between your estimation of the amount of adipose tissue and the amount of fat that the hot dog is purported to have. In addition, we also have a really neat spreadsheet in there that once you enter your data is already going to do the calculations for you and estimate the proportion of adipose tissue, muscle tissue, etc. It's even going to generate some pretty cool graphs that you can share with your class. And finally, there's a couple of research papers that have been done by scientists asking the exact same questions that we are today. That is, what is inside a hot dog? And these people actually went out, sectioned hot dogs, and then analyzed them in a manner similar to what we're doing today and actually published scientific papers on those. Okay, of those documents, the 2 things you're going to use the most are the histology scorecard we're going to use to score our hot dog sections, and there's also this cheat sheet in there that helps us to identify some of the tissues that we're going to see in our hot dogs. Because remember, these hot dogs have been profoundly changed. These are not tissues in their native state. The hot dogs have been cooked, they have mechanically separated meat in there, and so a lot of things aren't going to look like they do under the microscope in the lab. So let's take a look at some things we're going to see in the hot dog sections. First up here we have adipose tissue. Now, the adipose tissue in the left-hand side right there are actual adipocytes, but a lot of the adipose tissue that we're going to look at is actually going to be ruptured adipocytes, and so that lipid or triglyceride is just going to be floating free out in the matrix. So regardless of whether we see this or this, we're just going to call it adipose because it did come from adipose tissue. Now, probably the second most common thing you're going to see in your hot dogs is something that I'm going to call ground substance. Now, this isn't ground substance in the traditional sense of the word and that it's part of a connective tissue, but this is a artificially created ground substance. Remember, we cooked the hot dogs, we ground them up, and when we did that, we created this sort of collagenous goo that I'm just going to call ground substance. And so a lot of what we see in there, in between the adipocytes, is going to be this ground substance that is of an unknown nature. It probably is made up, again, of collagen and proteins and muscle fibers and things like that. Now, we might actually see some muscle fibers in there. So if you take a look at this section and this section, these are actual striated skeletal muscle. And we can identify them by their striations and the presence, if we can see them, of multiple nuclei. But unfortunately, when we cook it, a lot of the nuclei go bye-bye, Okay. Other things we can see in there, we might be able to see some blood vessels or possibly some nervous tissue as well. Not too sure about this, but this is my idea that I'm calling that nervous tissue, and it wouldn't be unheard of to find some nervous tissue in hot dogs. Just check that published paper that I have linked to. Other things we can see, hyaline cartilage. Hyaline cartilage actually goes remarkably well through the hot dog cooking process, and it's very easy to identify. We also have fibrous connective tissue, so anything that has visible fibers and seems to be an intact fibrous connective tissue, such as dense regular or dense irregular, we're just calling fibrous connective tissue. And then finally, you might see some bone spicules in there, and these spicules right there look to be of trabecular or spongy bone, so those are easy to identify. And finally, at the very, very bottom here, we have some really amazing sections, and these are all plant tissues. And we know they're plant tissues because we can see lots and lots of cell walls in there. You might be asking, what in the heck are plant tissues doing inside my hot dog? Well, keep in mind that some of these brands of hot dogs actually use onions and paprika and garlic and different types of vegetables and spices, and that these spices can actually show up underneath a microscope and they look pretty cool. Okay, now we're going to talk about how to analyze our histological sections and quantify how many different tissues are there. So this section right here is our third Vienna sausage section. And so you can see on the left-hand side or your right-hand side, this is a lot of hyaline cartilage over there, very easy to identify. All these big white droplets in there, those are all triglyceride resulting from adipose tissue. And then over on this side, we have some skeletal muscle fibers in there, so that's skeletal muscle. And everything else pretty much that we can see in there is that kind of ground substance material that was created when we took all those proteins and chopped up muscle fibers and we cooked them together at high temperature to give us this sort of artificial ground substance. And so that's what we're going to call it. So in order to quantify this, we need to lay a grid across. And here we go. We've got a grid, and this grid has 48 intersections. And so we're only going to identify the tissues that are under each intersection. And as we do that, we want to be going down there and writing things on our data entry sheet or our scorecard. Now, if you have two people, it works a little bit better. One person can be going from square to square to square and reading out their identifications, and the other person can be putting little hatch marks next to the appropriate tissue. Okay, since I'm here alone, I'm just going to work from left to right. So on the left-hand side, I can see that most of this in here, again, is hyaline cartilage. So I'm just going to go ahead and label all those points with a C. So far, 12 out of 48 points are cartilage. So far, this hot dog section is about 25% cartilage, which is kind of scary. Now let's look at the other tissues we can identify. Again, these big white droplets were adipose tissue or triglyceride. So let's go ahead and put an A over all the intersections that contain triglyceride or adipose tissue. Okay, so that's 9 points out of 48 points total. So far, this hot dog appears to be about 19% adipose tissue. Now let's look for muscle tissue. Now, even though we saw lots of muscle tissue over here, we only have one spot where there's actually an intersection with our grid, and so that one spot would be labeled as muscle tissue. So 1 out of 48 spots is muscle tissue, which is about 2% muscle, and that's not very much. But remember, this is only visible muscle. There's probably a lot more muscle in here that's just been denatured and broken down so much that we can't readily identify it. And so we have identified the cartilage, the adipose tissue, and the one muscle tissue. So what is everything else in this slide? Well, based on my analysis, I'm going to call everything else that sort of nebulous ground substance. And so we have 26 points out of a total of 48, or around 54% of the tissue here is identified as that nebulous ground substance. All right, so once you're done scoring each of the 12 different hot dog sections, you should end up with a score sheet that looks something like this. And you can see that we have little hatch marks for each different hot dog type and each tissue that we see in there. Now, we had 3 different subsamples for each hot dog, and each of those subsamples had 48 data points. So in theory, we should have 144 data points for each hot dog brand. The only reason we might not is if one of the sections, for example, has some area where there's no hot dog in the space that the grid is, and in that case, you're not going to count those data points. But for pretty much any of the other hot dogs, you're going to have about 144 total data points. Now, once you have those data points, you're then going to enter those into the spreadsheet that I've given you access to through the resources folder. Once you do that, you can generate some pretty cool graphs that look like this. So these graphs can show the proportion of different tissues found within our hot dog. And again, that's going to be our adipose tissue, our ground substance, the cartilage, etc. Now, these data aren't necessarily real data, but your graphs will look very much like these once you're done with your study. Okay, this concludes our introduction to the Hot Dog Histology Laboratory Exercise. If you have any questions, send me an email at langston@hawaii.edu. Meanwhile, happy hot dog hunting.